I'm going to continue with Oneness versus the 1% by Vandana, Shiva and Kartikey, Shiva. It is now more than 20 years since the miracle of so-called golden rice has been promoted as a cure for vitamin A deficiency. The last time the golden rice myth was resurrected was when Patrick Moore of Golden Rice Now was sent to Asia to promote the failed promise. Women across the world, organised as Diverse Women for Diversity, issued a declaration on International Women's Day 2015 that women and biodiversity feed the world, not corporations and GMOs. Bill Gates specifically set up the Cornell Alliance for Science to promote failed biotechnology by erasing the scientific evidence on the ground of the failure of GMOs and substituting it with propaganda pieces to attack local farmers, journalists and scientists as anti-science, to prevent real science and the reality of GMO failures from reaching the world. He finances a PR institute that masquerades as a scientific institution, originally endowed with $5.6 million by the Gates Foundation. In August 2019, the Cornell Alliance for Science receives a renewal contribution of $6.4 million in 2017. The new grant takes the total Gates contribution to $12 million. Cornell uses Mark Linus, a one-time vociferous opposer of GMOs, to fly into different countries to falsify the GMO story. Journalist and author Linus is new to the Cornell Alliance for Science and is sent to the deficient parts of the world to marginalise people's voices and put out the biotech industry's propaganda. When BT Brinjal failed in Bangladesh, he was sent to drown the local news of its failure through a planted story in the New York Times. When peasants distributed the GMO golden rice trials, disrupted the GMO golden rice trials in the Philippines, he was sent to muzzle the farmers' perspective and voices on GMOs. Gates and his expanding patent empire. Biopiracy of climate resilience in farmers' varieties. While talking innovation, Gates has engaged in piracy from software to seeds. At the Expo Milano 2015, during the Women's Forum organised by Emma Bonino, Italian's former foreign minister, I was invited to make the keynote address. In a panel following my address, a representative of the Gates Foundation talked of how it was financing the innovation and invention of climate resilient crops through new technologies. When I was asked him which farmers' varieties they were using, he was silent. Climate resilience is a complex trait and cannot be engineered through the crude tools of transferring single gene traits from one organism to another. What corporations and Gates Foundations are doing is taking farmers' varieties with known climate resilient traits from public gene banks, mapping their genome and taking out patents on the basis of guesswork and speculation about which parts of the genome contribute to the known trait. Along coastal areas in India, farmers have evolved flood-tolerant and salt-tolerant varieties of rice such as Bundi, Kalambang, Luna Bakada, Sankarachin, Nal Idhiluya, Ravna, Sayupadi and Dosh Sarakuda. Crops such as millet have been evolved from drought tolerance to provide food security in water-scarce regions and during water-scarce years. Navdanya decided to save the vanishing rice diversities of Odisha through a system of germplasm conservation, employing both in situ and ex situ methods, and at the same time carrying out experiments on their sustainability in varied ecoclimate conditions in view of rapid climate change and yield potentials under various soil and amendments. Their behaviour and responses are being recorded. This has come in handy while selecting the seeds of specific rice diversities for empowering local communities to rehabilitate agriculture in disaster areas like Erezama in Odisha after the super cyclone of 1999, Nagapattinam in Tamil Nadu after the Boxing Day tsunami in 2005 and Nandigram in Bengal in 2007. The tsunami waves affected agriculture due to the intrusion of seawater and deposit of sealand. More than 5,200 hectares of agricultural land in Nagapattinam were affected. The Navdanya team conducted a study in the impacted villages to facilitate agricultural recovery. We distributed 3 saline-resistant varieties of paddy, which included Bunde, Kalamambang, Ludabakda, to farmers in the worst affected areas. A total of 100 quintals were collected from Navdanya farmers in Odisha and transported over a distance of more than 1,500 kilometres from Balashore to Nagapattinam under the Seeds of Hope programme. The yield from these varieties were 3 times greater and better than those than any other known high yielders. The same varieties behaved even better when cultivated in Indonesia, another 1,000 or more kilometres away in 2006 by Professor Friedhelm Galton of Hohenheim University in Germany. Painting in life through genetic engineering is rapidly giving way to painting in life through mapping of genome. Navdanya Community Seed Bank in Odisha has conserved more than 800 rice varieties and multiplied and distributed salt-tolerant and flood-tolerant varieties wherever required. The innovation necessary for the evolution of these climate-resilient traits has occurred cumulatively and collectively over thousands of years. These traits and crops are commons, however, they are being presented as the innovation of scientists who renamed the flood-tolerant property in the farmers' varieties such as Dulaputi from Odisha as sub-1A or the submergence tolerance gene, using marker-assisted selections, not transgenics. Researchers were able to isolate the submergence tolerant gene sub-1A and then transfer it to a rice variety known as Swana that is grown on more than 5 million hectares in India and Bangladesh. Most rice varieties can tolerate flooding for only a few days, but researchers said that Swana sub-1 can withstand submergence for 2 weeks without affecting yields. This is a scientifically flawed claim based on genetic reductionism because flood tolerance, like other climate resilient traits such as salt tolerance and drought tolerance, are multigenetic traits. They cannot be identified as a sub-1A gene because it is not a gene. It has been referred to as a submergence tolerance 1, sub-1 quantitative trait locus QTL. What marker assisted selection does is to identify the genetic sequence that is already linked to varieties with the same varieties which share a trait. Such varieties are then selected for crossing conventionally with varieties like Swana. Farmers who bred the trait will not need market-orientated selections to breed for climate resilience. This is why we need to recognise the diversity and pluralism of knowledge system as well as diversity of language to describe the same processes and organisms. The agrochemical and biotech industry is using the climate resilient crops bred by farmers for their genome mapping, claiming the farmer bred traits as innovation through patents, but this is not breeding, it is piracy, biopiracy, and this is how the Gates Foundation redefines the biopiracy of flood tolerant rice from Indian farmers as an innovation funded by Gates Corporation has taken out more than 1,500 climate resilient patents on crops. In 2014, India's Patent Office rejected Monsanto's application for climate-resilient traits of cold tolerance, salt tolerance and drought tolerance in crop varieties evolved by farmers over millennia through applying their knowledge of breeding, while the spin is that these traits are being genetically engineered. In reality, it is biopiracy, unlike farmers who knew what they were doing when they selected and bred for specific traits. Corporations have no idea. Their genome mapping is done through computer programs and guesswork about which part of the genome contributes to which trait. The privateers of today's include not just the corporations, which are becoming fewer and larger through merger, but are also individuals like Bill Gates. Digital piracy of life, mining genetic data with DivSeek. DiversitySeek DivSeek is a global project launched in 2015 to map the genetic data of the present diversity of seeds held in gene banks. It is an extractive project to mine the data in the seed and to censor out the commons. The partnering organisation of DivSeek include the Consultative Group for International Agricultural Research, CGIAR centres, and public universities like Cornell and Iowa State, which are being increasingly privatised by the biotechnology industry and by Bill Gates. There are 7 million crops accessions in public seed banks. DivBank could enable only 5 corporations to own this diversity. Cornell is where the Gates Foundation funds the propaganda machine through the Cornell Alliance for Science. Iowa State University is where Gates funds the unethical human feeding trials of GMO bananas. The foundation also funds the partners of DivSeek, especially the African Agricultural Technology Foundation and an African-Brazil collaboration in DivSeek. This genetic colonialism is nothing but an enclosure of the genetic commons. A number of mass sequencing projects such as G2P-Sol, which plans to deep sequence 50,000 Solanaceae nightshade seeds, bean adept and 3,000 chickpea genome innovative are underway. The plants primarily originate in developing countries, especially in Latin America and Asia. Farmers' rights and the seeds they have evolved over millennia will be open to biopiracy through the genome database, just as maps were used to claim and steal territory in an earlier colonialism, and just as prior claims of communities were erased, all local laws ignored and violated. DivSeek is trying to bypass all access and benefit sharing provisions consistent with the Convention on Biological Diversity and, for some plants, the International Treaty on Plant Genetic Resources for Food and Agriculture, ITPGRFA. Key DivSeek members are inclined to ignore a resolution of the ITPGRFA adopted during the 6th session of its governing body in October 2015, asking them to report on the policy implications of DivSeek technologies, ignoring the rights of farmers, and treaties that protect biodiversity and create obligation for access. DivSeek is considering a Syngenta-inspired scheme to sell across to the sequence data of farmers' seeds from international gene banks. Evogene Limited, Israel, has patented a computer program for reading the plant genome. Its proprietary is silico gene discovery technology is called Athlete. In silico, as opposed to in vivo or in vitro, refers to investigations performed through the use of computer or computer simulation. Athlete is a computer database analysis program for finding gene function by comparing sequences from as many different plant species, tissues, organs and growth conditions as possible. Evergene claims its database has 8 million express sequences, 400,000 proprietary gene clusters and 30 plant species. The program clusters sequence according to a variety of criteria and then determine which gene candidate to investigate further. It is an informed winnowing process. ETC Group's report Capturing Climate Genes explains how. Athlete uses vast amounts of available genomic data, mostly public, to rapidly reach a reliable limited list of candidate key genes with high relevance to a target trait of choice. Allegorically, the athlete platform could be viewed as a machine that is able to choose 5 to 100 lottery tickets from amongst hundreds of thousands of tickets, with the high likelihood that the winning ticket will be included among them. Evergene also collaborates with Monsanto. A deal struck between the two companies gives Monsanto exclusive rights to a number of genes identified by Evergene that reportedly allows crops to maintain stable yields with lower applications of nitrogen. The companies collaborate on drought-tolerant traits as well. Monsanto and BASF, the world's third-ranking agrochemical company, are inventing $1.5 billion of collaborative R&D to develop high-yielding crops that are more tolerant to adverse environmental conditions such as drought. Like Athlete, DivSeek is based on the digital mapping of farmers' seeds, as Marie Hager of Digital Crop Diversity Trust says. Agricultural biodiversity is more important than ever, as it is the prerequisite for food security. By combining the information we have on where plants grow, information about their physical characteristics, and the sequencing data of the plant's DNA, we should be able to speed up the breeding processes. Whereas Cornell University Mays genetics Edward Buckler contends that the big issue of our times is the dematerialization of germ plasma. Germ plasma is becoming the information technology that it always was. Biopiracy is being addressed by laws that have been framed at international and national levels. It generally refers to the illegal appropriation of biological diversity and the traditional knowledge of local communities from developing countries by multinational corporations and research institutions. This definition mainly covers two aspects. One, access and use of biological materials and traditional knowledge without the authorization of the country of origin and the indigenous communities that hold and develop these resources, and or the lack of benefit sharing or unjust and unfair sharing of benefits with the countries and the communities who provide the resources. These forms of biopiracy are expressly prohibited by Article 15 and 8J of the Convention on Biological Diversity, CBD, 1992, and more recently the Nagoya Protocol on Access and Benefit Sharing, adopted by the Conference of the Parties to the CBD at its 10th meeting on the 29th of October 2010. At the international level, many developing countries have adopted legislation regulating access to their genetic resources and requiring benefit sharing in accordance with the principle of the CBD, such as India's Biological Diversity Act 2002. The country is in fact one of the 10 megadiverse countries in the world, according accounting for 7.8% of global recorded species, at a DivSeek seminar held from the 28th of November to the 1st of December 2016 at the Rockefeller Foundation Bellagio Centre in Italy. 18 experts discussed game-changing and potentially disruptive DNA sequencing technologies, big data platforms, high-performance computing, image-based phenotyping methods, gene editing techniques, and synthetic biology concepts, reveal the TWN briefing papers from 2016. So I will leave it there for now.